Hi, this is Dr. Goldkamp, and welcome back to the next episode of the Keto Naturopath. I'd like to continue where I left off last time. This is actually part 4 of how do we make epigenetic reverse aging work for you? And the question is, just in that title, is this marketing hype? Is this just a new thing you're pushing out there so people are going to buy whatever? Maybe a testing thing, maybe some supplements, maybe a facial, whatever. You know, it's not quite that way, but assume there is a degree of marketing hype in anything that can be marketed, okay? I'd like to boil it down to the essentials because it's actually very vital. And I spent the previous number of videos going over all this. I'd like to pick up where I left off because it's a cliffhanger if you think like me, all right? So let's get going. All right, so last time I talked about, we reviewed that some past studies, and I said, in order to understand the bigger picture, you really have to understand the history, how we came to this point. And I do believe that's the case. You really have to put a historical context around this to sort of say, wow, this is really pretty important, and each step of the way. So I just want to say, we started with the Dutch hunger winter in 44 to 45, then the siege of Stalingrad, St. Petersburg today, the Overkalic study. After that, right now, we are in the tail end of Dr. Randy Jirtle's studies with the agouti mice. And we want to explore a little more there because it really sets the stage for understanding the development of the Horvath clock or the epigenetic clocks that have developed since then, and how are they relevant, and what are they measuring, and how do we use them. Then there were some studies that came out, one we've already talked about, and then we'll get into actual testing. Okay, so let's get to it. So was Dr. Randy Jirtle's experiment no more than a parlor trick, turning the color of an agouti mouse from yellow to brown? And how is this connected to the human results of the study we've talked about before, potential reversal of epigenetic age using a diet and lifestyle intervention? And that came down to really boxing in men only for about 8 weeks to a healthy diet, saying this is what you have to eat, and potentially really one supplement. There's 2, but it's really just one. And a few other things, but primarily it was a whole foods diet. They're really amping up and saying, let food be your medicine, but with a critical eye of why this is relevant and what is in that food that possibly have made some men, nearly half got better. The majority just didn't change at all. And one got worse. How can that possibly be? Let's get to it. Okay, these are those results. 8 got better. They got younger, in essence, by up to about 10 years in the course of 8 weeks. That's phenomenal. And then some were just no change at all, and one got worse by over 10 years. So there you go. The average was 2.2 years for if you average all those people together. Okay, in order to use these results to increase your own health span, what we want to understand is the history of epigenetics, and for 3 reasons. We want to see how the effects are transgenerational. We want to know what is required to create an optimal methylation process, how to get tested, and why you'd want to do that, and why it's important. Picking up from that parlor trick, right? This is Dr. Randy Jirtle's. What he did, he changed a large obese, diabetic, yellow mice, and he did it many, many times, mice, prone to cancer, to normal colored, healthy mice. So they were the agouti mice because it's the gene that he's changing. So he gave them both mouse chow, and there really is a thing called mouse chow, and it's pretty standardized food, by the way, in the mouse world. But he gave one group methyl donors, pretty straightforward, B12, folate, SAMe, S-adenylmethionine, choline, and trimethylglycine, otherwise known as beta-ene. So both mouse chow and methyl donors, keeping it simple. So this is what happened. Poof. The younger generation came out normal. They had they were not prone to cancer or any sort of diseases. They did not share the same color as their mother. And what happened? All right, so back in 2000, this is from that article I just referenced. Back in 2000, a professor, Dr. Randy Jirtle, was a professor of radiology oncology at Duke University, which is actually just up the road here. And his postdoctoral student, Robert Waterland, designed a groundbreaking genetic experiment that was simplicity itself. Really interesting, he's a radiation oncologist. They started with pairs of fat yellow mice known to scientists as agouti mice. They're called agouti mice because they carry a particular gene, the agouti gene, that in addition to making the rodents ravenous and yellow, renders them prone to cancer and diabetes. Sounds like the American public, huh? This experiment was designed to see if they could change the unfortunate genetic legacy of these creatures. Typically, when agouti mice breed, most of the offspring are identical to the parents. So this is a mouse that has been bred specifically to be generation after generation after generation after generation, if it's crossed with another agouti mice, to always be obese, diabetic, prone to disease, and yellow. Always. So that goes back thousands of generations, probably even more. They're so certain of this. They're genetically identical. Typically, when the agouti mice breed, most of the offspring are identical to their parents, just as yellow, fat, pincushions, and susceptible to life-shortening diseases. The parent mice in this experiment produced a majority of offspring that looked altogether different. The pups, as they're called, were slender and mousy brown. Moreover, they did not display the parent's susceptibility to cancer and diabetes and lived to a spry old age. I don't know how old mice are when they get spry or old. The effects of the agouti gene has been virtually erased because of what they did in this experiment. It was accomplished without altering a single letter of the DNAs, mouse DNAs. So they didn't change the genes. They changed this thing called the epigenome. Basically, they changed the directions of what genes will be turned on and what genes will be turned off. That's the epigenome. All right, this approach was radically straightforward. They changed the mom's diet starting just before conception. So not like generations and generations, just before conception. They fed a group of mother mice a diet rich in methyl donors, which are components of the methylation cycle that produce methyl groups. Okay. Methyl groups are small chemical clusters that can attach to a gene and turn it on and off. Shazam! That's what they got. A healthy, normal mouse. Question: how is that different than what honeybees do? Is the difference due to being methylated or about having different methylation patterns? So being methylated is to have all genes turned off. So I'm saying, did the mouse experiment, the Dr. Jurtles experiment, simply turn off genes? Or did it enable a healthier methylation pattern, which was what that other study was about, trying to establish a healthy methylation pattern, keeping the things that are supposed to be off, and the genes that are supposed to be on, and helping create a functional, healthy mouse on. Okay, queen bee to worker bee. All right, so why is this similar? Kind of similar. It's what they were fed that changed the outcome. So you have a bee larvae, which is genetically identical. So we're not even, this is better than the identical twins experiment. From scratch, identical larvae. And from scratch, they were fed two different sources of food. One, of course, for the queen, future queens, is called royal jelly. But for the worker bee gets pollen and honey and a thing called bee bread, which is actually a mixture of the pollen. But the important thing is here, it comes from plants. So you have one that came in through the door, right? Came in through the door with the pollen, and they made the honey of it, and they gave it to the kids, gave it to the larvae. All right, well, it's just like Randy Jurtles. He gave them both mouse chow, and he had one special addition to one group of mothers. So the queens and workers are alternative forms of adult female honeybees. And I don't know if you knew that. So it's all female we're talking about. So one's fed the royal jelly, the other's fed the pollen and honey. Bee bread, which is what they call it in the biz. Okay, queen bee lives for 5 years. The worker bee lives for 40 days. And their difference is completely due to their different diets. So it's the power of diet. Don't lose that. It's the power of diet. All right, so the question becomes, what's the difference between royal jelly and bee bread, pollen, and honey? Okay, let's get to it. Plant-based is, so the pollen and the honey, so the pollen's attached to their legs, the honey is what they make in the hive. It's all plant-based. And there's a thing in there, in this plant-based, all the phytochemicals, a family called flavonoids, and specifically a subset of that called phenolics. And they really boiled it down to one of the phenolics, one of the flavonoids, coumarric acid, which is prevalent in honey and bee bread, regulates genes involved with a caste, right? This is a caste determination, inhibits ovarian development in the worker bees. So all the worker bees are females castrated, if you want to use that. They have no functioning ovaries. It's been permanently turned off, not just once, but it's on a regular basis. So they developed without any ovarian support, no reproductive function at all. The genes are turned off. But with the royal jelly side, that's pretty interesting. It's not plant-based. So it didn't come from anything that walked into the door. It came from, actually, the worker bees produce a secretion. In the biz, they call it bee snot, if you want to know. But it's secretions that are fed into the royal jelly. It's produced from secretions of the worker honeybees. It promotes ovarian development, of course, because this is where all the eggs come from. And it blocks any effect of the coumarric acid. So what it does, it prevents, if there is any contamination, if there's an off day and somebody got confused and gave the future queen bee a regular bee bread, pollen and honey, well, this has anti-coumarric acid prevention there. So not only does it prevent from any accidents from happening and having any effect on the future queen, but it also has the bee snot, the royal jelly, that helps produce and stimulate ovarian development, obviously for the egg. So it turns on specific genes, and here it turns off specific genes in the same set. So there's nothing magical about the DNA. That set, and it even, there's so many different studies on this now that it even goes down to slight brain different development of what genes are turned on in the worker bee and not turned on in the queen bee. Okay, so not easy being a queen bee, as you probably can guess. There are numerous behavioral, physiological, anatomical differences between the queens and sterile workers, even though they are identical at the genetic level. Upon emergence from the pupa, the new queen engages in a series of duels with rival queens, and perhaps you know some of this. The single survivor will leave the hive for 1 to 5 mating flights, during which she visits sharply delineated leks. That's what they're called. There's males out there waiting for just this purpose in their life. Congregations area used solely for mating that may be several kilometers from the hive, where hundreds of drones typically await to try to mate with the queen. Queens will mate with an average of 12 drones, who die shortly afterwards. The mated queen returns to her native hive. Egg laying begins shortly afterwards, and she will typically not leave the colony again unless a new queen is raised in the subsequent year, in which the old queen leaves the hive with a large swarm of workers and relocates to a new home. So you heard when a hive swarms, you see they all follow the queen. They're trying to protect the queen. All right, so what does this have to do with anything? Let's go a little bit further. So the queen's retinue, so when she's laying eggs and so on and so forth, here's what's happening. They feed the queen, right? They feed the queen, they constantly groom her and they lick her, and in the process, they're picking up the queen's mandibular pheromone, which suppresses their ovarian development. So here's the queen that has ovarian development on steroids, so to say, suppressing everybody else's ovarian development. Remember, these are genetically same. So if you took any of these bees and looked at their genes and go, wow, they're the same. How did they turn out so different? This is the point. Okay, why did Randy Jirtle's experiment create these results? He gave them methyl donors. So methyl donors, B12, folic acid, SAMe, choline, trimethylglycine. These are supplements that you can get Amazon, that we have in our pharmacy. They've always been there. But as a focused collection of supplements, they have a power that is, for some people, not for everybody, they have the power to methylate. Complete the methylation for some people. Okay, so to the agouti mice, these supplements turned off specific genes. It methylated them. It silenced them. Didn't kill them. This completely changed their methylation capacity. So it gave them a lot of methylation factors to make more methylation, more methyl groups, which then can be passed on primarily by acetyl-methionine to all of their genes to turn them off. So they gave the substance that was needed in order to make more methyl groups So that's their capacity, methyl capacity, i.e. their ability to produce methyl groups, which then change their methylation pattern. Their pattern is what genes are turned off by being methylated, what genes are left on. So their ability to control what genes are turned off or on, which restored their normal function. So it's remarkable. You have this genetic mouse, the agouti mouse that has been specially bred for this to be fat, heavy, diabetic, prone to cancer, and yellow. And they changed all that in one generation. Poof, changed all that in one generation. Pretty unsophisticated in the sense of there wasn't a lot of intervention. Here, eat this. Here, eat this. So these methyl donors are found in food, human food, bee food. So I gave them both chow, right? You've seen that before. Okay then. And he got the normal mouse back. Technically, this is called a pseudo-agouti mouse because the DNA didn't change, but how those genes were used changed. That's your epigenome was changed. Hope you're getting that differentiation. So it created a different methylation pattern, arguably a healthier. It defaulted to this healthier pattern. All right, function was restored. I use a piano to sort of look at all the genes. So if you had playing a piano and some keys didn't work, you just couldn't play what you wanted to play. You fix all that by giving it methyl factors, all the keys work, and then you can play Beethoven or Schubert or whatever you want to play. Okay, here's what happened. Now we're looking a little deeper here. So this is called the methylation cycle. Primarily all of this is considered that, with an emphasis really right here, the methionine cycle. You've seen this before in past videos that I've done, but this is your factory. This is your methylation factory. It pops out these little ping pong balls called methyl groups that get stuck onto the genes that your methylation pattern, however we don't know how this is determined, turns off genes that shouldn't be on. And this is one of the functions that we lose as we get older. Suddenly we can't keep certain genes off. So certain oncogenes, cancer genes, tend to suddenly be on with time. And that's not a good thing. So you have a number of these genes and processes that should be silenced. I've also referred to this as the glutathione factory. And I want you to think about this pretty much as an assembly line or as a series of traffic circles, which I've used before, interlinking the fact that they all relate to each other. So the Krebs cycle, otherwise known as uric acid cycle or TCA cycle, is really about how you make your energy. And I'm just showing that it's related. I'm not going to get into that. But these are neurotransmitters. This is your folate and B12, very important. There's MTHFR. So this is what determines your ability to methylate your genes, your methylation capacity. Okay, think of these as interconnected assembly lines, right? So if you're Elon Musk and you're making your cars, you want to make sure you have all the things that are necessary before you start the process so you can pop it out on the other side. Methylation, methyl groups are given off right here from SAMe. It's called the universal methyl donor. Okay, here's more of a very general neurotransmitter synthesis, folate cycle, methionine cycle, and what they call transsulfuration cycle, because glutathione is a sulfur compound. You know, cysteine is a sulfur compound. That's why you're told to have your sulfur foods so you can make glutathione. So whether you want to think of this of your glutathione factory or your methyl factory, that's fine. They both suit my purpose. Okay, what did methyl donors have to do with all this? Well, Randy Yertle, he basically plugged in these specific, there's your choline, there's your B12, there's your B9, and there's your trimethylglycine. All of these now make sure that there's plenty of methyl groups to kick off, primarily by SAMe, goes off. So these are ordinary supplements. Here's two other studies, in case you think this was, oh, that was way back, how 2003 and 2007-ish. No, this is still cutting edge. So here's a study that you can go look up if you're interested. Early life nutrition and mental health. Did you know those who don't have enough methylating factors have certainly a higher rate of things like spina bifida, cleft lip, but schizophrenia, major depressive episodes, bipolar, obsessive compulsive disorder, basically psychiatric mental disorders are very high. Autism also. Autism is multi-factor, but this is a part of it. And here's another one. Methyl donor micronutrients, call them cofactors, call them vitamins, if you will. But all these things we've been talking about that modify DNA methylation in cancer outcomes. In other words, they're looking at those genes. I just said that as we get older, genes like oncogenes just start doing bad things. They start hyperplasia and so on and so forth. And how do you control that? Well, now they're looking into controlling the methylation cycle of these genes to turn them back off, to silence them. They shouldn't have been on, they should be silenced. And this is just another way of drawing the drawings that I've shown you before. It's their way of doing it. So there's your SAMe, there's your SA, accidental homocysteine, there's your MTHFR. We're going to get into it a little bit. Okay, I'm just saying that this is another way of using this, of looking at it. It is very useful in so many different contexts. And there's another way. So there's your methionine cycle, your folate cycle, and your what they call biopterin, or your neurotransmitter cycle. All these dark blue boxes are actually enzymes, and they are places in which it's common to have a mutation. Now, they're not all, some are your mutations make them speed up too fast. They work too fast. So if you think of that assembly line, like Elon Musk's big factory, like somebody's doing too much, it's like, well, it doesn't help with the next step down. And, but anyway, so these are pretty common in my world, enzymes that you check out. You find out who has whatever mutations for these. They're not all of equal weight, by the way. Certainly MTHFR, methyl tetrahydrofolate reductase, and a few others, methionine synthase and methionine synthase reductase. Who cares? Big names, right? And they're SAMe. These are all problematic genes with a certain mutation. Not automatically problematic genes, but if you have a mutation, it can be a big problem. It will suddenly slow things down. If you slow things down here, you slow down both sides. Okay, here's another way of looking at it. These are what I considered the initial 3 problem genes that I look at. There's MTHFR, MTRR, which is methionine, CBS. And so what do you look for? We'll get into the human application later with blood work and so on and so forth, but I want you oriented to, this isn't Greek. This looks a little bit, oh my gosh, I'm not going to look at it anymore. This directly has to do with DNA methylation. DNA methylation is turning genes off. You methylate, you turn it off. So this whole little methylation factory makes the supply of methyl groups to be in abundance. So when you need to turn off a gene, you can do that. It doesn't automatically turn on. So as we get older, we start losing the ability to turn off genes. And so what happens? They start to activate, like the oncogenes. Okay then, so the end result is the experiments show that you can give vitamins, otherwise known as cofactors or micronutrients, that will replenish or increase one's methylation capacity, ability to produce methyl groups. The agouti mouse model has demonstrated the importance of a maternal nutrition in shaping the epigenome, right, the ability to turn genes on or off, of the offspring. The abundance of methyl donors in the maternal diet, such as folic acid, accidental methionine, B12, choline, trimethylglycine, determines the methylation of the agouti mouse, which in turn renders the mouse coat yellow when not being methylated enough, hypomethylated. So yellow is hypomethylated, and it turns it brown or normal color or healthy color when it's methylated. Okay, so 8 years later, Dr. Randy Jertle does this again. 8 years later. I mean, it is still cutting edge. It's like they couldn't believe it. Of course, it was done all over the world. It was such a simple experiment that anybody could do it. So they did it again, and he did one change though. So there's the reference to that. I'm going to get into it. Same outcome, but he changed it. He added a known toxin. And this known toxin is called BPA, Bisphenol A. It's in all the plastic bottles and so on and so forth. A little bit of a divergence here. I say BPA is still a concern. It was a known toxin back in the 70s. So it's always an issue. The plastic bottle is always an issue. The plasticizers and hospitals and so on and so forth, always an issue. Cellophane, it's definitely rich. It's all these sources. But just want to give you a glance at 4 different studies, I think actually 5, that we got 19 gene expression and DNA methylation changes in parts of the brain due to bisphenol A. Bisphenol A induced epigenetic changes in male reproductive systems. This is in humans. Prenatal bisphenol A exposure linked to epigenetic changes in glutamate, in rats. DNA methylome, which is basically the area that gets methylated. Wide alterations associated with estrogen receptors dependent effects of bisphenol A, because in this context, bisphenol A, those plasticizers, are considered a xenoestrogen, an artificial estrogen. They can link to estrogen receptors and start a lot of craziness, right? Okay, and the last one is genetic and epigenetic alterations induced by bisphenol A exposure during different periods of spermatogenesis. This goes back to male hypo or greater hypofertility, or the increase of male infertility has gotten to be really, really high. I haven't covered that as a topic. It's pretty interesting, but this is one of the reasons xenoestrogens, chemical estrogens that come through our food affect spermatogenesis in men. Okay, anyways, back to Dr. Randigudi. So he took the large, obese, diabetic yellow mouse prone to cancer and chronic disease. He gave them both the toxin. Gave them chow, so they're having a good life. They're having their mouse chow, standardized mouse chow. Must get boring after a while, I think. BPA. But with the BPA, he then gave the same methyl donors. So what do we get? All right, same results. The pups were normal. Wait a minute. Let's go a little deeper. Here's the analysis on that. So when you have just the chow, when you just have the mouse chow, that middle group, that's the yellow you're looking at. There's a yellow and there's actually a reddish in the middle. So that's the control group. And so when you add dietary, the methyl donors, or genistein, hadn't talked about that, that's from soy, what do you get? You get all normal brown mice. What happens when you add the BPA with no methyl donors? Well, now they're all yellow. They stay diseased. They stay obese, diabetic, and prone to cancer. Okay? Now you give them the BPA, and you already know that they're going to stay prone to cancer. You give the BPA, bisphenol A, and now you give the methyl donors, or genistein. What happens? They become healthy. Okay, so environmental epigenetic studies. Variable yellow agouti mice. So now you have this mouse, the agouti mouse. Now he realized, wow, you can get all shades from yellow to brown by what you give them. So here's what he did. He gave the methyl donors, you saw that, so they became hypermethylated and they end up being brown. He gave them genistein. Genistein is from soy. It's also in tofu, and it's also used as a botanical medicine in certain aspects. Highly estrogenic, by the way. So that also would be considered a xenoestrogen, a phytoestrogen, if you will. And so they ended up being hypermethylated, and they're brown. You go, wait a minute, I thought this is all these problems. It's a plant, anyway, it also stimulated, without it being the methylation factors, the same outcome. Also, alcohol created the same outcome. So how can you explain these here? Well, part of the explanation is, as he's explained, that I'm not going to put this down here, it stimulates the body to, wait a minute, we have an emergency, let's see if we can compensate. So there's enough reserve in your methylation capacity to compensate for these insults. The genistein will stimulate it. Maybe not forever, the ethanol will stimulate it. So obviously an alcoholic is going to fall apart eventually, right? So it doesn't work long term, but a little alcohol, and he puts a little asterisk then, and they probably researched as to how much. Bisphenol A gets the hypomethylated. So there's yellow. So if it's brown, here, if they're yellow, down here. So hyper and hypomethylated, those are the concerns. Those are the things that I want you to take out of this. Hypermethylated, hypomethylated. So how is any of this relevant to what we've been talking about? This is actually big time relevant, because here is the shift I'm hoping your perspective will take at this point. You are the agouti mouse. I am the agouti mouse. We are now in a new era of living as humans, in which we do have toxins in our environment that never existed before. Bisphenol A is just a good example of one, and that's been around since the 70s. We have plenty of other toxins. We also have a whole era of foods that's devoid of nutrition. I'll call it the Dorito diet. These are things you can eat for caloric gain, but they don't have all the micronutrients that are required for us to operate correctly. So where do the wheels come off the car? The wheels come off the car exactly here. This is how it's coming off the car. It starts specifically with this methylation factor, if you will, this methylation system, our methylation capacity. We are losing our ability to methylate on a discretionary basis. What genes should be turned off? We are aging earlier. You can say, well, wait a minute. I thought our longevity is going up. Well, different topic. It actually has come down. But when you think of what the agouti mouse is and was, it is obese, it is diabetic, it is prone to cancer and other diseases, chronic diseases. And how is our culture? I'm speaking in the US, our current rate of chronic disease is 60%. That means regardless of age, 60% of our population has at least one chronic disease. If you get over 50 and it just gets worse, obviously. But that's huge, considering 1935, it was only 7.5%. What changed things? So this issue, so we're talking about epigenetic aging and reversing aging, you could say we're making ourselves just healthier. But this is how we have to focus on it, and this is why it is so acutely important to understand what is going on when we test ourselves for epigenetic age reversal. This is it. Up next, what does the epigenetic age test do and how does it connect to this? Sort of told you a little bit of that. How do people's micronutrient levels affect that? Call them their nutrition levels, call it their vitamin levels, however you want to look at it. It's basically we need to have those cofactors to make the factory work correctly. There can't be any absenteeism. We need to have that not in excess. We need to have that on a regular basis because a lot of water-soluble vitamins, if that's what we want to call them, can't be supplied in excess. And we can't just have a lot of the fat-soluble vitamins, but we need to have an appropriate amount on a regular basis. It's not a bank account. So how do people's genetics, single nuclear polymorphism tests or analysis affect this? These are the things we're going to be looking at. We sort of peeked a little bit of that, and I'll give you a peek here now. This is what we're looking at. These are just 3 of the many genes we looked at, 3 of the many enzymes that have genes that we can analyze. And these are the more prominent ones that do cause a lot of problems. So back to the first study we talked about how to make people younger. That group that benefited were the ones that had problems with their methylation cycle. Hi, this is Dr. Goldkamp again. I just wanted to say, if you're interested in the topic of epigenetics, you may well want to start here and go through some of the videos we did on this. I think it's a profound new tool that we all can use. So until next time, take care.